Hello students, a very good afternoon to everybody. So first of all, before we start our today's session, okay, I'm going to launch some polls for technical check. Okay, please let me know if my audio and video is clear. And for the people who are on YouTube live, you can put it in the chat box if there is any technical problems. Excellent. All right. So I end the poll. Okay. So I'll just cancel it now. Okay, fine. So yesterday we had very interesting conversation, right? So we were talking about Mendelian inheritance, like how non-Mendelian inheritance and what are the examples of non-Mendelian inheritance. Okay. So today, anyway, we'll pick up from the last class and we'll discuss a little more on chromosomal theory of inheritance. Okay. So if you had any doubts from yesterday's class, you can just put me on the chat box, okay, so that I will check up and I will answer if the time permits. All right, fine then. So let's move on to our first slide. Hold on. Yeah, sorry. Yeah. So our first question, right? Anyways, we have to take a quiz before the class. So the first question is here: ABO blood group system is due to. So we studied about blood grouping, right? And we studied what are the factors affecting the blood grouping, like how the blood groups, represent themselves in phenotypic characteristics. So here are our option: multi-factor inheritance, incomplete dominance, multiple allelism, and epistasis. So here is your options. So I expect you to answer within a minute. All right. Okay, I'm launching a poll. Try to understand the question. Yes. Come on, few more people. Please try to answer. Yes. Perfect. So we are 30 seconds up and you have given your answers as C. Okay. All of you have given your answer as C. So why C? So we know that multi-factor inheritance and first of all, multi-factor inheritance is completely different. Incomplete dominance, we did not study in ABO blood typing. So multiple allelism, since we have IA, IB, and small I as three alleles that is responsible for phenotypes of our human blood groups. That is why our option is C, isn't it? Okay. All right. So our next question. Yeah, this is an interesting question. Look here, three children in a family have blood groups O, AB, and B respectively. What are the genotypes of their parents? Okay, so here are the given genotypes. So I give you 1 minute for you to answer this question. Okay. Okay, 1 second. Yeah. So here is your poll. You can answer this. Yesterday we talked about this. Yes. So we have one answer within 30 seconds. That's excellent. Come on, students, we have 15 seconds left. All right. So we reached 1 minute and I'm going to end the poll right now. Okay. I think it's a fair decision. So many of you answered, I mean, like the people who got the answer right was B. One of you answered B. Okay. So the answer is B actually. So let us see why they got the B. First, we'll try with option B and then we'll see the other options. Okay. So we have one parent with AB blood group. So we'll write it as IA and IB. Okay. And then other parent is having O blood group. So we'll write it as I and I, right? So now the offspring should be IAI, and then we have IAI, and then we have, yeah, IBI, and then we have IBI. So is this the correct option? We have A, and then we have B and B. So now you know that it is not the correct option. Sorry. Okay. Our first option, IA small I, sorry. IA small I. Wait, let me erase this. 1 second. Let me erase this. Okay. IAI. And then we have IBI, right? So this is our first option. So if I take IA, IB, and then I have IA and small I, and then I have IB and small I, and then I have small I and small I. So we have the possible blood groups as AB, and then here we have A, and then here we have B, and then here we have O. See, they have asked about A, B, and O, isn't it? So in this case, the people who have taken option B, right, we have only A and B blood groups. So we have got here A, B as well as O. So they have mentioned about 3 offsprings, isn't it? So we don't worry about the offspring which is not born yet. Okay. So we have this option. So let us see what are the other options we have. So I'll erase this. Okay. So we can have space for workout. All right. So I'll erase your option also. Yeah. So let us see other possibilities from the given options. IB is one parent, and then we have IA and IA as another parent. So obviously IA, IB, and then we have IA, IB, and then we have IB, IA, IB. Again, we have IA, IB, isn't it? See, if you take it like this, okay. So it's a little messy, but we can make it as quicker as possible. So we have all the blood groups as AB type. You see here AB, 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 and AB. All right. So our option C is wrong and our option B is wrong. Okay. So let's go for D. IA, IA, and then we have IB and small I. So what are the possibilities? IA, IB, IA, small I, and then we have IA, IB, and then we have IA and small I, isn't it? So we have AB blood group and we have A blood group. See, AB, AB, and we have A and A. So our right option is option A, isn't it, people? So did you understand this? Is it clear? Please respond. So anyways, I can skip this part. Okay, so you let me know if you did not understand. So let's go for today's topic, that is chromosomal theory of inheritance. So we were talking about this, isn't it? So after the discovery of meiosis, which is one of the revolutionary thing happened, so people started understanding more and more about the importance of chromosomes in heredity, right? So when Walter Sutton and Theodore Bouvier, so they started working, okay, when they started working on different organisms, they understood that chromosomes really play an important role in carrying the characteristics, and that was possible by understanding cell division, isn't it? So what happens in most of the organisms, since we are in diploid state, so meiosis is one such cell division that happens where our characteristics will get transferred from our parents. For example, if you have a mother and father produces gametes like sperms, and it has only haploid number of chromosomes, that is 23, but the father's somatic body will have 46 chromosomes, isn't it? So now when the chromosomes are segregated during your meiosis, that plays a very vital role in carrying the characteristics from one generation to another generation. Now, why chromosomal theory of inheritance is very, very important? So one of the reasons is, you remember when we talked about Mendelism. right? When we talked about Mendelism and how Mendel's ideology of inheritance was not well accepted, right? One of the reasons was what? Because of the diversity that we have in our population. Take human beings or any other organisms that are present in our environment. They don't always come with contrasting characteristics like tall, dwarf, black, white, or something like that. We have mixed traits. Okay, and Mendel could not prove it. So chromosomal theory of inheritance is one insight that will give more and more evidence linking with Mendelian genetics, and it gives, it will just spread the sheet. It is not telling that Mendel is wrong. They're telling Mendel is right in many cases, but there are also other possibilities where even chromosomal behavior is responsible for carrying the characteristics. Okay. So now when it comes to chromosomes, yesterday we talked about what are homologous pair of chromosomes and how they duplicate, right? So here is one of the best examples of how the chromosomes segregate very independently, right? So if we have two cases, right, we have two sets of chromosomes in, let us say this is set A and this is set B. So you can see here where the orange chromosomes, like where the orange chromosomes are going, orange is going with green here, and again orange is going with red here. So there are two possibilities in which what characteristics are chosen and how these chromosomes get segregated during the meiosis. Okay, so this independent assortment, like the way this chromosome segregate independently, so that is the reason why we cannot predict the outcome. Okay, only when the child is born we can understand what are the characteristics that are transferred from parent to an offsprings, or what are the new characteristics that the offspring has apart from the parents. Okay, so now what we have to understand here, chromosomes function as vehicles of heredity like that of your genes, and genes are present on the chromosome. That is why the movement of chromosomes matters a lot in carrying the hereditary information. That is the second point. And third point, whenever we see a chromosome, for example, 1 and 1, chromosome 1 and 1 or 2 and 2, we call this as homologous pair. And whenever this 1 and 1 and 2 and 2, 3 and 3, 4 and 4, like this, so on. So when they segregate, right, during meiosis, they segregate independently. So this is what these scientists were trying to conclude. But for the chromosomal theory of inheritance, so one of the very pioneers of understanding, one of the main scientists who gave us a very elaborate understanding of how chromosomal inheritance, okay, happens, or what influences the chromosomal inheritance, that was given by Thomas Morgan Hunt. So usually whenever we have fruits in our home, we can see the small flies, right? So we call them fruit flies or Drosophila melanogaster. So he chose Drosophila melanogaster as an ideal, okay, experimental organism to conduct his experiments. So one of the main important aspects of genetics and molecular biology or genetic engineering, you take anything with respect to genetics, okay? For us, model organisms become very important criteria. We can't take just human beings and start experimenting. Why? Because we have to look at so many different factors. For example, if I'm working under laboratory conditions, I would require, okay, I need to distinguish sexually reproducing organisms very easily. For example, if you take the male Drosophila and female Drosophila, you can see that the male is quite smaller than the female, right? So morphologically, the male is small and the female is big. So this is very identifiable character. Okay. So this is one thing. And the second thing is the number of chromosomes on the Drosophila is very, very few. Okay. It's very less number of chromosomes. So it is very easy for us to understand what genes and where they are present and how they are responsible for a particular character. So that is the second one, is a gene size. And the third one is the variants. Okay. So if I'm working on a genetic module, so I need different population. I need a population study so I can understand variation. I can understand the similarities in a very elaborate manner, isn't it? So the more the variants I can have, the more it will, enhance my studies for analysis. So I can understand how this is different from the others and what are the similar characteristics that are between these two. And next one is, because of the size of the Drosophila, it is so helpful for us and it is so useful for us, and it is very convenient for us to grow them in a very large number in a synthetic medium. Okay, so in a very short span of time, that is in 12 to 14 days, they will finish their life cycle. So within 12 to 14 days, we will have their offsprings, and the offsprings characteristics can be studied compared to that of the parents. So these things will happen very, very fastly. So that is why I told you model of an organism is very, very essential for us in molecular biology or genetics. See, take in humans, okay? Imagine how long it would take for a human or a cat or a dog or any other bigger organism to reproduce and create variants. And it is not ethical also, right? So that is why this is a small organism. Life cycle is very, very short. Okay, it can be easily distinguishable. A number of offsprings, hundreds of offsprings can be grown within a short period of time in a synthetic media within the laboratory. Okay, so this is why we can say that Drosophila was chosen as a model organism for many of the genetic studies. Okay, so we even call Drosophila Cinderella of genetics and studies of genetics. Okay, so fine. Next one. So the concept of chromosomal theory of inheritance, it can be understood very well clear by these two words, that is linkage and recombination. So first of all, whenever we talk about alleles or genes, okay, so these alleles and genes, where are they present? So we know that they are present on the chromosome. So whenever they are present on the chromosome, they are present on the same loci of a chromosome. Same loci means, look at here, A and B are, see how they are present, A and B and A and B. So they are present at a very similar distance and very similar region on the chromosome. That we call it as loci of a chromosome. Okay. So when they are present, in a loci of a chromosome, now what happens is the distance between the two genes. Let us say that the A is one gene and B is another gene. Okay. So now the distance between the presence of A gene and B gene matters a lot. Okay, in how the offsprings will get these characteristics in the next generation. For example, if I tell, in this case, you can see that tightly linked they have given, sorry, I will just erase this. Okay, they have given this as tightly linked. Okay, tightly linked means what? Look at these gene pairs, how closely they are present with respect to each other, right? So similarly, when you look the same genes now or different genes, so look at their position, they are quite far from each other compared to, compared to the relative positions. So this is called as linkage. So linkage by definition is called as physical association of the genes present on the same loci of a chromosome. So physical association in the sense they are not attached to each other based on how near or how far they are present. Now this inheritance or transfer of characteristics quite matter. Are you understanding? Is it clear? Please let me know if my pace of class or my language, or if you have any problems, please put it in the chat box so we can correct it in the next class. Otherwise, I don't want you to have any problems with respect to class. All right. So fine. As I'll keep moving on because we have very short time. You just let me know. Okay, so this is about physical association of two genes on the chromosome. So whenever they are associated physically, now what happens is whenever the chromosomes get segregated, right? We know that in meiosis we have one of the important process that is called as chromosome crossing over. That is overlapping of arms of chromosomes, of different arms of chromosomes happen during crossing over. So whenever the crossing over process happens, right? Now what happens? Bulk transfer of genes takes place from one chromosome to another chromosome. What is bulk transfer? Bulk transfer is, for example, this entire segment of the gene can be transferred to the one chromosome. Okay, this entire gene when it overlaps with this. Similarly, here also, only this portion might be going to the other chromosome, something like this. Okay, so now what happens? Homologous chromosome, when they align, and when they cross over, now what happens when they cross over? Bulk transfer of genetic material happens. Whenever there is bulk exchange of information is happening between the chromosomes, now that will result in what? Recombinant chromatids. What is recombinant chromatids? Parents had these character, Now the offspring's chromosome, whatever the sperms or the ova that are going to be produced, right? Now they will have either this character or this character. Okay, a special character. So this we call it as recombinant characters. So now to understand linkage and recombination very clearly, okay, so let us see what Morgan did. So Morgan selected Drosophila, right? So when he selected Drosophila, he conducts independently two different crosses. One is cross 1, other one is cross 2. Okay, so when I tell cross 1 and cross 2, these are independent crosses. Okay, so they are not relating with each other. So in the cross ones, he selects the following characteristics. What is, see, in cross 1 he selects these two. Okay, so what is that cross 1? What is he concerned about in cross 1? In cross 1 he selects, see here, yellow bodied, white-eyed females. You can see here the body is yellow and you have a white eye, right? So you have a white eye here and you have an yellow body, and this is a female. And then he chooses brown body, right? Brown body. And then what is the eye color? Red color eye. And what is this? This is a male. Okay, so we have white eye, and then we have yellow body female, and we have red eye and brown body male. So this is in cross 1, and this is the first cross. And in the second cross, what he chooses different criteria. What is that? The chosen characteristics for these crosses: white-eyed miniature wing. Miniature means small. Okay, now you can see here you have white eyes and the wing size is relatively small compared to this, right? You have a longer wing here. So whenever you have a long wing, the opposite of long is miniature or small. Okay, and then we have the other organism, that is red eye and large wings. You can see here it has a red eye and it has a very large wing. So cross 1 is yellow body, white eye, brown body, red eye, and cross 2 is white eye, miniature wing, and then we have red eye, long wings. Now when the cross is conducted, what we can understand is, now in the cross A, when we talked about the body color and the eye color, look at the genes. Basically, these genes are present on the X chromosome. Okay, so that is why in males here in the Y chromosome, this is Y chromosome, and there is no genes present on the Y chromosome. All right, so it is present on the X chromosome. What is present on the X chromosome? The genes that are responsible for eye color, okay, body color, and then wing size. All of them are present on the X chromosome only. Now understand the relative positions of this. See, the eye color, okay, the body color and the eye color are very closely present to each other, right? So they're very close to each other. Similarly, when you look at in cross P, when we talk about eye color and the wing size, look at the distance of the gene present on the same chromosome, okay, on the chromosome of same loci, okay. So when you look at this distance and see what happens with cross 1, after F1 generation, okay, in F1 generation, he finds that, most of them are having parental characters here and some of them are exhibiting a little bit of recombinant characters, okay. Now he goes for cross 2. When he conducts cross 2, now he gets a certain percentage. What is that percentage? When you analyze the population of F2 generation, okay? So now, since we are taking two crosses, we call it as dihybrid cross, right? Di means two, hybrid means characters. So since in the dihybrid cross, according to Mendel, we have to get 9:3:3:1 ratio. But here we don't get any 9:3:3:1 ratio. Whenever a mathematical ratio deviates, that means that it is not following a particular rule, right? It's as simple as that. But they did not reject this idea of 9:3:3:1, but they went ahead to understand what was really happening here. Now, what happened here, since the genes were very closely they're very closely present with respect to each other, with respect to color of eye and body color. Now, what happens is in the F2 generation, among 100% of the offsprings, 98.7% of the individuals were having parental type. I'll explain you what is parental type, okay? And only 1.3% of the individuals, they were showing recombinant type, okay? Whereas when you compare it with cross B, what do you get? You get only 62.8% of the population of the F2 generation were showing parental type, whereas the recombinant type were 37.2% were recombinant type. Now, what can we conclude out of this? So what is parental characteristics? Imagine when you are at home and then you have exact height, exact skin color, and exact eye color, and exact hair type as your father or mother. So whenever the some of the characteristics exactly match your parents, that we call it as parental characteristics. Okay. Now, what is recombinant characteristics? Sometimes we assume, okay, my mom and dad are fair, but I'm dark. So now I'm assuming, like, how am I dark? That is called as recombinant character. The new characteristics that are shown or that are expressed in a offspring, okay, of the next generations, that we call it as recombinant characteristics. Now, what can we tell about this? So linkage is responsible for recombination. More the linkage, less is the recombination. Less the linkage, more is the recombination. You can just tell with respect to proportionality. Okay, I'm very poor at maths, so we can just understand by this, right? So more linkage, less recombination, and then we have less linkage, more recombination. As simple as that. Okay, so here the position, the relative position of the alleles was really influencing how the characteristics are transmitted from one generation to another generation. Okay, fine. So next one. A very important aspect of this experiment is, after this recombination concept was more popularized, we have Alfred Stewart. Okay, so what he did was he understood, based on the recombination frequency, right? We can take two characteristics, two different characteristics, and understand how much recombination are they promoting. For example, if I tell body color and eye color, look at the relative position, they're very close to each other. So when you look at this closeness, they promote only 8% of the recombination. Whereas when you look at this, the eye color and the wing size, they can promote up to 9%. That means the distance is larger between eye color and the wing size. Okay, when you compare between the body color and the wing size, they were promoting up to how many percent of recombination? 17% of recombination. So this is called as, this aspect of mapping the genes based on the recombination and the linkage is called as physical mapping of genes. Okay, so in the next chapter, when we study about molecular genetics, okay, or molecular biology, there we can understand the importance of this gene mapping. Okay, and how it influenced in one of the mega projects that was ever conducted, that was HGP. Okay, so this is biologist greatest achievement, we can tell, HGP. Okay, one of the mega projects across the entire globe. So we'll study more and more about HGP in the coming sessions. Okay, so this is about gene mapping, and that was about Drosophila, recombination, and linkage. Okay, so if you have any doubts, please let me know now. And the YouTube guys, please, you can put it in the chat box so that we can clear your doubts. Okay, if that's fine. So can you give me a reaction at least, like if you have understood the session or something like that, so that I feel confident that class is going well. Okay, so now our next topic, the part 6 of this chapter, is sex determination. So we are a little bit aware about sex determination, right? So what is sex determination? Knowing the sex of an individual, okay, is called a sex determination. And we studied the same sex determination even in while studying human reproduction, right? Reproductive health. Where there we understood, okay, we can determine the sex of a baby. Okay, basically the sex determination can happen in many ways. Okay, we can do by physical determination. By looking at an individual, we can understand whether it is male or female. For example, in humans, if you look at humans, we can understand whether a person is male or female, right? So similar to that, we have way too many types of sex determination: environmental based sex determination, autosome based sex determination, sex chromosome based sex determination, and so on. But in today's session, we will start talking about, okay, autosome and number of chromosome, as well as sex chromosome sex determination. And overall, what we're talking about, chromosomal based sex determination. So usually what happens is in today's genetics, we follow a technique called as karyotyping. So karyotyping means what happens. See, for a healthy man, we have a chromosome 1 to chromosome 46. We have its relative length, the bands, okay, the size, and the ratio of this chromosome is very fixed, right? So now we can compare this healthy individual to different people in the population. Now, if there is any alteration in the chromosome number, shape, size, or structure, we can understand, okay, there is some difference between individuals. So this study, which helps us in understanding the chromosomes, it is called as karyotyping. And how did it start actually? So Helking in 1891, he started understanding about spermatogenesis, that is production of sperms. While he was studying spermatogenesis, what he came across is 50% of the sperms received a particular nuclear structure and 50% of the sperms did not receive it. Okay. So whenever he was studying more basically on insects, whenever insects you see, in insects you have XXXO type. Okay. So whenever we have XXXO type, now some of the sperms received this X body. Okay. Some of them did not receive anything. Okay. No, nothing. I'll just write it as NO. So he named these sperms, whoever got this X, okay, as X body. He just called it as X body. And today we call it as X chromosome. And that is the discovery of the first sex chromosome, we can call it as. Okay. So let us see what are the different bases on which we can determine the sex of an individual. First one, we talk about is XX and XO type of sex determination. So before we go into the XX and XO type of sex determination, first we have to understand, and female also produce gametes, right? So whenever female produce gametes, let us say that the female produces XX. Okay, the female is XX, right? So whenever female produces gametes, all the eggs that she produces will have X, only X. Whereas male, if I tell male, okay, if I tell male, human males, let us say that we have, let us say no, we have XY chromosomes. Okay, so when we produce sperms, we have X sperms and we have Y sperms. So this one, whenever an organism produce uniform type of gametes, we call it as homogamety. Okay, this we call it as homogametic. Whenever we have a different gametes, for example, male produces X sperms and it produces Y sperms, right? So that we call it as heterogametic. Okay, now you understood the term called as homogametic and heterogametic. Now let us go for what is XX and XO type of sex determination. First of all, where can we find this XX and XO type of sex determination? This type of sex determination, you can find it in most of the insects. Okay, in most of the insects, such as grasshopper, you have cockroaches. Okay, so these organisms exhibit this. So in these organisms, you can find here the male. Okay, the male here in insects, they have only X chromosome and they do not have any type of other chromosome, other sex chromosome. Whereas the female, okay, the female has two X chromosomes. Now, what happens when the gametogenesis happens? The male produces X type of sperms and O type of sperms, that is without any sex chromosomes. Whereas the female produces X type of chromosome and X type of chromosome. Now, when they mate, if X sperm goes with X egg, okay, I will write it as egg. Here I will write it as sperm. Okay, sorry, I'm making it as congested. Okay, so X and X goes, here we have XX. Again, X and X goes, we have XX. And here we have XO, and then we have XO. Now you can see here, all these will become females and all these will become male. So since the male produces two types of gametes, what are the two types of gametes? One sperm will have X chromosome and the other sperm will have no chromosome, right? Only O, only or nothing will be there. So since male produces two different types of gametes, we call this as male heterogametic. Okay, and the female produces only one type of gametes, that is only X type of gametes, that we call it as female heterogametic. Okay, did you understand the XX and XO mechanism? Okay, and in our next thing, we are going to study about XX and XY type of sex determination, which is very, very popular, right? So in humans, it happens. And even in Drosophila, it is XX and XY. Just now we studied about linkage and recombination. And I told you that the Y chromosome do not have any of these genes. So you remember that we have both humans as well as your Drosophila. They exhibit this XX and XY type of sex determination. There are many organisms. For our example, now we are taking about humans and Drosophila. Okay. So now what is XX and XY? The female has XX chromosome and the male have XY type of chromosome, right? So whenever we produce eggs, so when a female produces eggs, okay, she will produce uniform type of eggs. That means all the haploid eggs will contain only X chromosome. Whereas when you talk about male will have 50% will have X chromosome and 50% will have Y chromosome. So this condition again will take us where? Male heterogametic condition, isn't it? Now look here. If this sperm, I will write it as sperm here. Okay. So now here, look here. If X sperm goes with X egg, then we'll have XX. That is a female baby is born. And then we have X sperm, X egg going with XY sperm, then we have XY. Then again, X sperm going with X egg, we have XX. And again, we have XY. Okay. So now we understood very, very important concept. What is that? In our society, we blame a lot on female, right, for not giving the male child. It's been happening for a very long time and it's still happening. So basically the female is abused or harassed for not giving a male baby. But now we know scientifically who is responsible for producing male babies. So that is male. Why? Because male produces two different types of gametes, isn't it? So it depends on him what type of sperm will go and mate, okay, with what type of egg. That will determine the sex of a baby. Okay, so this is XX and XY type of sex determination. Okay, so now interesting example. So in female, especially when it comes to birds, okay, so birds are very peculiar example. Why it is a very unique example is because if we look at birds, okay, the birds have male gametes of ZZ type, okay, like how we had in female XX. Similarly, the male birds will have ZZ type of sex chromosome, whereas the female, okay, she will exhibit ZW type of sex chromosome. That means what? Whenever a male produces, he produces 100% of the sperms of same type. Only Z type of sperms will be produced. Okay. Now, when it comes to female, the female produce 50% of the eggs will be of Z type. Okay. I'll write a 50%. And then W type will be again 50%. Isn't it, people? So now female is produced two different types of gametes. Now we call it as female heterogametic. Okay, whenever we talk about where do you find this female heterogametic condition of sex determination, we have to directly think about birds. So in female birds, she will produce two different types of eggs. One is of Z type, other one is of W type. Whereas the male produces only uniform type of gametes, that is Z and Z. That is why we call female has heterogametic with respect to birds. Now we can understand how the babies are born. So let us say that the ZZ is male. Yeah, it is male actually. ZW is female. I'll write it as female. So whenever she produces, now ZZ goes again, it produces male, again ZW female, again ZZ male, and ZW female, male, male, female, female. Okay, so this is how the offsprings are born. Okay, I hope you understood this particular concept with respect to ZZ and ZW and most of the birds. Okay, now only in birds we can blame females, right? So why didn't you give this particular baby? Not in humans. Okay, so the female is responsible here for giving rise to what kind of baby. It's all chance factor. It's very natural. We cannot predict the outcome, okay, when it is happening naturally. Anyways, now another interesting example. This is called as haplodiplontic type of sex determination. Okay, so haplodiplontic, haploid and diploid, okay, number of chromosomes will determine the sex of an individual. Very interesting, isn't it? So when it comes to sex determination in honeybees, okay, we have females, that is the queen bee, and then we have drones, which is the male bee, right? So when we look at this male and the female, okay, now their number of chromosomes are very, very, different. What is that? Always the female is diploid, whereas the male is haploid. Okay, the female will have 32 chromosomes, that is the diploid set of chromosomes, whereas the male will have haploid or half the set of chromosomes compared to that of females. Okay, now what happens? Now, whenever we look at the gametogenesis process, first we have to talk about how the gametes are produced, isn't it? So whenever the female produce gametes, that is eggs, she has to undergo reductional division, that is meiosis, right? So she had 32 chromosomes, so she goes under reductional division and she will produce what? 16 and 16 eggs, all right? So she will produce eggs having haploid number of chromosomes. Whereas male already have only haploid number of chromosomes. When he undergoes gametogenesis, now what happens to him? Can he go under meiosis? Definitely no, right? He cannot reduce the chromosome number from there. It's not possible. So now what happens? The male undergo, hold on people. Okay, where is it? Where is the cursor? Okay, the male undergo mitosis. When male undergo mitosis, he will produce, okay, the sperms which are containing 16 chromosomes. Okay, so let us see if I have a slide. Okay, yeah. Yeah. So now what happens? 1 second, let me find some space. Yeah, I'll write it here. Let us say that we have egg. Okay, 16 and 16 are eggs. Okay, and then we have 16 as sperms. Okay, 16 as sperms. Now remember, this is a sperm and these are eggs. Okay, these are eggs. Now, if the sperm gets to meet the egg, now what will happen? This will result in a zygote containing 32 chromosomes, right? So when it results in a zygote containing 32 chromosomes, that will result in female. Okay, whereas what happens to this egg which does not meet the sperms? This egg will undergo parthenogenesis. Okay, that is fertilization. Production of offsprings without fertilization. So when it undergoes parthenogenesis, now this haploid number of egg, whatever that was there, that will develop into male B. Okay, so this kind of, okay, this type of sex determination based on the number of chromosomes, this we call it as haplodiplontic sex determination. Is it clear, people? Do you have any doubts with respect to haplodiplontic? Please let me know. Okay, I will move ahead. Okay, so you can just put a message. Fine. Our next part is mutations. Okay, so when we talk about mutations, so whenever you hear a term called mutations, suddenly we either remember some supernatural powers, or some ways we will think that, okay, it is really, really bad. Okay, when we take the examples of Hiroshima and Nagasaki, okay, or Chernobyl disaster, that is nuclear disasters that has happened. So whenever we talk about how mutations are terribly wrong. So whenever we talk about Deadpool, X-Men movies, okay, so we talk about, oh my God, it's so cool. I wish I was a mutant, okay, and so that I get supernatural powers, right? So we assume mutation in a different sense, right? But when it comes to natural environment, mutations play a very key role. So they are very, very important for the survival of entire biosystem. How is it even possible? So mutations are supposed to be bad, right? So when we talk about the changes that has to happen with respect to an organism, right? In an organism with respect to its environment keeps changing, its food habits keep changing, and the earth's climate, everything keeps changing. If you track the history of earth, so the earth has been changing, very gradually, but it is keep on changing, right? So if an animal or an organism fails to adapt to these new environmental conditions or new conditions wherever it lives or whatever, with respect to whatever it is, now the survival of this organism is impossible. So that is when the mutation and recombination comes in hand. Okay, so mutation and recombination are two important tools of evolution. So mutations are, sudden changes that happens in a genotype of an organism. So usually we have one specific pattern of genes. So our genes are basically codes, right? So there is a sequence of genetic codes starting from one point to another point. So that we call it as gene. So whenever this sequence is altered suddenly or deleted, or the reading frame is shifted, so that we call it as mutations, and that happens on a very sudden basis. So whenever we talk about mutations, the mutations can be of two types. Okay, one is point mutation, other one is frame shift mutation. So let's see what are this point mutation and frame shift mutation. When it comes to point mutation, there is a possibility whenever there is a single base pair change, okay, only one nucleotide change. See, we call it as nucleotides. We see the original sequence here. So this is thymine, adenine, adenine, cytosine, thymine, guanine, cytosine, adenine, ATCG. You know about this. If you don't know, you will learn even more elaborate in the next chapter. There is no problem, okay? So this is the original reading frame. You know that here is T, see here is T, and compare this with here, TT, AA, AA, CC. And here instead of T, what is here? C is there. So whenever there is a single nucleotide change, okay, in the reading sequence, okay, that can be either removed and replaced by another type of a base pair, that will lead to point mutations. And point mutations of several types, okay? So it can be silent mutations, it can be nonsense mutations, or it can be missense mutations. What is silent mutation? So usually, for example, let us take here, okay? Now you can see here TTC, okay, and AAG, here TTT and AAA has resulted in, okay? So now whenever you compare this sequence, it will give rise to a same amino acid, okay? So even though there is a change in the base pair, but at what amino acid has to be there, it will be the same amino acid. So basically the protein function, see what happens, the DNA is converted to RNA, the DNA messages in the DNA or the gene is converted to mRNA, and the mRNA messages are used to construct proteins. And this is how all our biological expressions, all the genetic expressions happens. Now, whenever there is a change in the nucleotide in the DNA sequence, it will affect the RNA sequence. Whenever there is an effect in the RNA sequence, it will affect in the protein building, isn't it? If you are not given a right construction plan, then it will lead to some problems. Exactly, right? So here also the same thing is happening. Now, what is happening here? They are talking about one type of nucleotides. Instead of T, the A is replaced, even though there is, instead of C, there is T is replaced. So even though the T is replaced, but it will result in the same amino acid, you can see here. So this kind of mutations, there is a change in the base pair, but yet there is no change in the function of proteins. It has not affected the function of proteins. So this we call it as silent mutations, okay? Now, next one is nonsense mutations. What are nonsense mutations? Whenever we talk about genetic codes, there are certain codes which do not talk about amino acids. See, we know that proteins are built by amino acids. So whenever there are no codes, whenever there are codes that is not asking for any amino acids, so that we call it as nonsense codes or stop codes, okay? So when it results in the stop codon, we call it as nonsense mutations, nonsense point mutation, we'll call it as. So what are nonsense point mutations? Whenever there is a change in a gene sequence, that will result in what? Creating no amino acid. There is no amino acid added in the sequence. So usually that doesn't happen. So whenever there is no amino acid, or the sequence is telling there is no amino acid, so that will lead to what? Nonsense point mutations or stop point mutations, okay? Next one is missense mutations. So when we talk about missense and missense mutations, there are two types of mutations. One is conservative and non-conservative mutations. So when it comes to conservative mutations, now what happens? So there is only single base pair change. So when it results in single base pair change with respect to conservative mutations, okay? So whenever there is a single base pair change, the addition of amino acid, whatever the amino acid that is being added, okay, because of the change, that amino acid will have very similar properties of the original amino acid with respect to its polarity, okay, with respect to its functional group, okay, with respect to whether it is ionic, okay. So based on, we have chemical properties, right? So based on the chemical properties, even though there is a different amino acid that has been added, okay, but it will have a similar, okay, chemical characteristics. Usually conservative point mutations are not that effective. whereas compared to that of non-conservative mutations. When it comes to non-conservative mutations, a completely different nature of amino acid is added, okay, in the non-conservative thing. Whenever there is addition of an amino acid which is completely irrelevant, okay, now what happens? Just because of a single amino acid insertion, okay, just because of a different amino acid insertion, now the entire functionality of the protein changes. It will not function as original proteins, okay. So that is the problem with non-conservative mutations. Non-conservative mutations are quite deadly. They will tremendously lead to loss of functions. Whereas in conservative, what happens? In conservative mutations, it's quite different. They can enhance the protein functions. That is one thing that can happen here. Okay. So we understood point mutations. Point mutations can be of several types: silent, nonsense, missense. Okay. Silent one is there is a change, but we cannot understand. Okay, because since there is a same amino acid added. Nonsense is because of the, see, UAG, UAG is a stop codon. It's a code that says there is no amino acid. So since there is, it's not asking for any amino acid, that we call it as nonsense codes or nonsense point mutations. Missense, conservative or non-conservative. Conservative, a similar property of amino acid is added that results in enhancement of protein function. When it comes to non-conservative, now what happens? There can be a possibility of tremendous loss of protein function, or an entire different protein can be synthesized that will result in severe genetic, okay, expressions. Okay, so one of the examples of point mutation is sickle cell anemia, and we will be talking about it in detail. Okay, the next type of mutation is frame shift mutation. So whenever we talk about frame shift mutation, here you can see in the left end, you can see there is a reading frame, CAT, 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 cat. So this is the actual reading frame. So this is how we read. We start from one point and we keep on going to the other point. So this is a normal gene sequence, okay? Now, when you look at a frame shift mutation, now what happens is the entire reading frame is affected because of either deletion or insertion or duplication. We'll talk about it. See, let us say that BEAST, B-E-A-S-T, BEAST is a normal reading sequence. I go from B-E-A-S-T. So this is how I read. Now what happens here? Instead of B, F is substituted. Okay, that will lead to what? Feast. Now the reading frame is changed. Now the entire meaning of the gene sequence also changed. Or look here, between B and E, if I add R, that will become breast, right? Again, the meaning is different and the reading frame is different, and that we call it as insertion. All right. So now if I delete some sequences, what are the deletion? If I delete A out of this beast, now that will become best. Okay. Now, if I invert a sequence, okay, BEAST was there. Instead of ST, I'll make it as TS. So now you can see that the entire reading frame has affected, isn't it? So whenever there is an entire reading frame affected, the entire meaning of the gene and the following mRNA and how it has to produce the protein, everything will change. Okay. So that is called as frame shift mutation. And that is due to substitution, insertion, deletion, inversion, etc. Okay. So when we talk about mutations happening at the genetic level, that is the gene level, we can talk about frame shift mutations, or when we can talk about point mutations. Whereas whenever we talk about higher mutations with respect to chromosome itself, you know, the chromosome can carry several hundreds and thousands of genes within one chromosome. So whenever there is a problem with a loss of a fragment of a chromosome, or additional fragment of a chromosome is added, okay, or an entire chromosome is deleted out of the sets of chromosome, that we call it as chromosomal aberrations. Okay. So whenever there is chromosomal aberration, it leads to severity in genetic expressions. One of the examples is cancer cells. Okay. So this is about mutations. Okay. So what causes mutations? So again, when we talk about mutations, it can be so many different types of mutagens, isn't it? For example, it can be physical, chemical, or biological. So any three can cause mutations. So any agents that can cause mutations, we call it as mutagens. Okay, mutagens are responsible for causing mutations. So if we talk about physical mutations, we talked about, a nuclear blast or nuclear radiation escape. So we know that the nuclear particles or radioactive material, they emit radiation, and the radiation comes in the form of rays, that is alpha, beta, and gamma radiations, right? So whenever these radiations actually penetrate our cells, now what happens is, if you have a particular sequence in your genome, let us say that this you have thymine. Okay, thymine is T is one of the sequence, isn't it? If a UV rays falls on thymine, now what happens is, instead of having a linear DNA, now your DNA becomes like this, something like this. So instead of one thymine, you will have two thymines. Now the entire structure of the gene is affected, isn't it? So now this we call it as thymine dimerism. That is because of UV radiation. That's how we get skin cancer. So alpha, beta, and gamma radiations are very, very, very severe. They affect a lot of genes at a lot of points. Okay, and same goes with X-rays. Okay, whereas chemical mutagens, it can be of so many types: mustard gas, phenol, formalin, acetic acid, formic acid, and ammonia. They can all cause mutations, and they can cause alteration in the genes that can result in abnormalities in physical expressions. Okay, so this is about mutagens. The agents which cause mutations are called as mutagens, and there are of two types: physical mutagens and chemical mutagens. All right. Okay. So now we have to go to pedigree analysis. Since I have only 5 minutes of the time, I will stop here. Okay. So we can continue in the next class. So before you go, I have a small request. Okay. Just give me a second. Give me a second. I will actually launch an NPS form. Okay. I think it's launched. Yeah. Please let me know since you are not talking. Okay. Okay. Here. Okay. Sorry. Yeah. Here is your form. All right. Sorry. Here is your form. You can just give a review on the following questions. By the time you finish the poll, I will just check on the YouTube. Okay. Is the poll gone? I don't know. Okay. Let me do it again. Yes. Okay. You are answering already. So I assume we have 6 people, including me. Oh no, only 3. Okay, I think I don't know who has answered. There is one more JNV Mahe and JNV B 12th, right? So I don't know who has answered. One more thing, one more answer has to come from either of the classes. So I will wait for your approval. Okay. All right, people, I will assume that your answers are, it makes me happy to get these answers. Okay. Thank you very much. And I will meet you in the next class in the next weekend, and we can continue our concepts and we can discuss even more better. Okay. So you can leave the session. So